My name is Sydney Waterman,
And I'm Megan Walker.
and this is our video project. So our learning objective is to explain why studies of yeast can lead to insights into the biology of human cancers. So we wanted to define what a yeast was. So yeasts are single-celled fungi, and they're found on plants, leaves and flowers, soil and saltwater, and on skin and in human intestinal tracts. They also can reproduce by budding instead of binary fission, which is shown in this picture. You can see a yeast is budding basically into another yeast. And so we wanted to provide a few examples of yeasts. So here's an example. It's called Saccharomyces pombe, and it's a fission yeast with rapid generation time of around 2 to 4 hours. And it's a model of cell cycle control because it has a distinct G2 phase, and this allows scientists to study its cell control and how it works. And here's a picture of it. To the right, it's blue. And here's another example. It's Saccharomyces cerevisiae, and it's a simple and easy to grow budding yeast. It has a high frequency of nonhomologous recombination, which can give rise to chromosome repairments and DNA repair pathways. It's also used as a model for cell cycle control and for transcriptional regulation, and it allows scientists to help us study yeasts and cancer in humans. And here's an example of a picture of it to the right.
So we also wanted to give a definition for cancer, and that is a term for diseases in which abnormal cells divide without control and can invade nearby tissues. So the cell division cycle is shown in the image, and it is controlled by the cell control system, which signals cell division or blocks cell division. And when it has a mutation or it is failing, it causes that uncontrolled cell growth that causes cancer in humans. So yeast cells can be used to help study human cancer because there are a lot of similarities. The proteins used in the cell cycle in yeast and the cell cycle in humans are very similar. They both rely on cyclin-dependent kinases and phosphatases. So the mutations that inactivate cell signals in yeast cells correlate to the mutations that cause mutations in the cell cycle in human cells. And this image is just showing how cyclins are involved in the cell cycle. So yeast cells help us study cancer because the similarities in the cell cycle between yeast and human cells allow scientists to study uncontrolled cell division that causes cancer in humans by experimenting on yeast cells. And one of those studies is was conducted by Lee Hartwell, who was a baker's yeast or studied baker's yeast, and Paul Nurse, who studied fission yeast. So they studied a mutation in a gene that coded for the cyclin-dependent kinases, and when that gene was mutated, the cell cycle was stuck or misbehaved, and the cell was not duplicating properly. And because that gene is very similar in humans as it is in yeast, that mutant explained what causes the rapid cell division in humans that leads to cancer. So that is just one example of how studying yeast can lead to insights on studying human cancer. And here's our works cited. Thank you for watching.